Hi everyone, this is our presentation on imputing DNA sequences using KNN and Levenshtein distance metrics. The sequencing of genomes has helped advance the field of genetics and biology by enabling scientists to gain an accurate understanding of organisms. However, a major problem in this field is that DNA sequences are easily degraded, and when that happens, the data gets lost. The limited accuracy of current sequencing methods prevents the formation of reliable conclusions since they are both slow and inaccurate. Our research aims to use machine learning, specifically KNN, to impute or fill in these missing values.
So our model uses Cannon algorithm or k-nearest neighbors. So the average of the closest k-neighbors is taken to make predictions. So the prediction for x, so on the right, we have a Cannon example. So the prediction for x when k equals 3 neighbors is b, because b is the most frequent value out of those 3 neighbors. And the prediction for x when k equals 7 neighbors is a, because a is the most frequent value out of those 7 neighbors. So our approach combines KNN with the Levenshtein distance metric. So Levenshtein distance is used in place of metrics like Euclidean or Manhattan distance. Levenshtein distance measures how similar two strings are by counting insertions, deletions, and substitutions required to transform one string into another. So on the right, we have a example. So we have C, T, C, G, C, C, and T, A, C, G, C, G. So we start off by deleting the C in the start, insert a after the T, and substitute the last C for a G. So there are 3 total steps to transform sequence 1 into sequence 2. So the Levenshtein distance should be 3. So Mus musculus was chosen as our model organism because the complete genome is known. So phylogenetic analysis revealed Mus pahari and Mus caroli to be most similar to Mus musculus from available data sets. So we use Mus pahari and Mus caroli to impute Mus musculus's DNA. So our method could also be used to impute other species' DNA.
So to get the data for the moose genus, we went to a source called NCBI, and we downloaded the data for the genomic data in FASTA format, which is just a standard format for genomic sequences. So originally we downloaded the data for each chromosome, but the files were way too large. So we switched to downloading for each gene. And as you can see on the right, there were around 14,000 genes in common between the 3 species, and only a very small proportion were not in common. But we were only able to test on a subset because there was so much data. And once we got the data, we just did some pre-processing. So because Mus musculus, their genome was completely known, there were no values that were actually missing. So there was nothing that we had to impute. So we had to mimic missing values. And to do that, we would start at a random index I in the genome. And then from there, we would generate a random number L. And then the next L characters would be masked. And then those are the values that we would try to impute.
Now let us see how the algorithm actually works. So we have this flowchart here, which we'll be following in order to see step by step how we actually predict values. So let's take an example. I have a Mus musculus sample gene and a Mus caroli sample gene. We'll start by randomly masking sequences of lengths varying between 3 to 8 in the Mus musculus sample gene. As we can see here, we've masked 2 different sequences of lengths 5 and 6. We'll first start by imputing the value for the first blank length. So the next step is to compare the sequences before and after the blank space. So over here, as you can see in the Mus musculus sample, we've taken a sequence of characters before the blank space as well as a sequence of characters after the blank space, which we will be comparing to the Mus caroli sample to get predictions. We'll compare this to every single possible set in which we have 20 characters before the blank space and 20 characters after the blank space in the caroli sample, because we've set the value as 20, which can be changed later on. As we can see over here, this is the first possible such set with TGAAG as the prediction, and the yellow and orange subsequences representing the sequences before and after. We take the Levenshtein distance between these sequences, as we can see over here, and then we average them out to get the Levenshtein distance for this particular prediction. We then move on to the next iteration in the program, in which, as we can see over here, we have skipped a character, and then we do the same thing. Just the difference is that the prediction over here is GAAGT, and we take 20 characters before the prediction, 20 characters after, get the Levenshtein distances, and average them out. The model does that for every single such prediction until we've got a number of predictions from which we will decide what the final prediction is. We're doing this for a few predictions in this example just to get an idea. As we can see, we're moving characters forward, and now we've got predictions.
So once we have the final neighbors list with all the Levenshtein distances for each prediction, we sort them in increasing order. And once they're sorted, we take the first k neighbors, which in this case k equals 3, with the lowest Levenshtein distance. And then we choose the most common prediction, but since all the 3 predictions in this case were different, we just choose the one with the least Levenshtein distance, which is the first one. And then finally, we calculate the accuracy. And so for the first prediction, the accuracy was off by 1 base pair. So the accuracy was 80%. And then we ran the simulation again for the second blank space and got an accuracy of 83.3%. So the total average was 81.8%.
Now that we've seen how the algorithm actually works, let's look at some of the accuracy metrics and hyperparameter tuning optimizations that were implemented. So there were 3 methods that were used to measure accuracy. The first one being percent full matches. In this was a more of a binary method in which we would check if the entire prediction matched the correct values. That is, every single base pair matched those in the correct value, and only then would the prediction be termed correct. We soon realized that this was not very optimal, as it did not give a better idea of how accurate our program was. So we shifted to a percent base pairs correct method, in which we analyzed individual base pairs and their accuracies. So if 4 out of 5 base pairs in a prediction were correct, the accuracy would be 80%. We'll average all these accuracies and get the final accuracy. The average normalized Levenshtein error was the third metric, which was basically the average of the Levenshtein distance between the predictions and their correct values. Now, we also implemented some methods to improve computational efficiency and predictive accuracy. The first one being punishing insertions and deletions. Early on during analyzing the predictions, we soon realized that insertions and deletions could actually affect the accuracy of our predictions by quite a lot, which is why we decided to punish their contribution to the Levenshtein distance and thus improve accuracies. The second method was introducing a maximum possible difference between consecutive neighbors. That is, for any given neighbor, if the Levenshtein distance of that neighbor exceeded that of the previous neighbor by more than a set value, then we would ignore that particular neighbor and every other neighbor after that one in a sorted array of neighbors. We also implemented a method that skipped iterations. As explained in the algorithm design, we were moving 1 character forward in every iteration. But we realized that if we moved a multiple number of characters when certain conditions are met, we could save on computational power. The fourth and final hyperparameter tuning optimization that was used was a score cutoff, which basically capped the maximum possible value of the Levenshtein distance. And if during the calculation of the Levenshtein distance, the value exceeded that of the maximum possible value, the calculation was stopped then and there.
Initially, the search area was set to 20 characters, which meant the algorithm compared 20 characters before and after missing sequences in the model organism Mus musculus to corresponding sequences in the similar species, namely Mus caroli and Mus pahari. So why did we end up using 20 characters? Well, we initially performed tests to determine the optimal value for search area, and as you can see in figure 9, which is a graph of search area versus accuracy peaked at close to 88.1% using a search area of 20 characters and dipped beyond this point. To see if our model would generalize to higher lengths, we also tested the model at varying base pair values between 0 and 1000. As shown in figure 10, which is a graph of maximum blank length versus accuracy, the algorithm was shown to impute with over 70% accuracy at lengths up to 300 missing base pairs and declined beyond 300 characters. The 3 remaining tests we carried out were testing imputation using multiple species versus a single species, punishing insertion and deletion of characters in Levenshtein distance calculation, and using k-neighbor pruning. These tests were used on 5 randomly selected genes in which hundreds of predictions were made. As shown in Table 1, accuracy increased significantly with the use of multiple species from 76% when only using 1 species to 84% for 2 species. With the addition of an extra weight for insertions and deletions, accuracy further increased by another 1%. And lastly, by implementing k-neighbor pruning, accuracy further increased by 3% with 2 species and an insertion punishment. The 2 main strategies we used for improving computational efficiency were implementing a score cutoff and skipping sequences with high Levenshtein distance. As seen in Table 2, we tested the runtimes using both of these strategies on 5 randomly selected genes and found that a score cutoff had little impact on computational efficiency, whereas skipping increased computational efficiency by nearly 25% by decreasing runtime. And the combination of skipping and a score cutoff yielded similar results to the use of no cutoff or skipping.
In many experimental analyses and studies, missing DNA base pair values have prevented scientists and researchers from drawing accurate conclusions and results. Current imputation methods are prone to data degradation and are thus unreliable. A new approach based on KNN algorithms, Levenshtein metrics, and phylogenetic analysis of similar species can impute missing real-world data values while maintaining high accuracy. Instead of implementing pre-existing algorithms, this new method for DNA imputation can significantly contribute to the systematic evaluation of many genomes. Our results have shown that using 3 species in the KNN calculation, punishing insertions and deletions, and pruning k-neighbors resulted in the most accurate predictions. In future works, we plan to find a way to optimize the computational efficiency. This way, we can not only make use of our improved algorithm to predict DNA base pair values with high efficiency, but also do it in a computationally less intensive way. We also plan to run extensive tests on a higher number of genes and use the same model to check accuracies on species similar to each other belong to other genera.
We would like to thank NCBI for providing us with the genomic datasets for the moose genus, and also like to thank Max Bachman for writing the efficient Levenshtein distance library that was used. And lastly, we would like to thank our mentors for their continuous support and guidance. These were the references that were used in this particular presentation, and we thank you all for listening.